today I'll be talking about the development of a new risk of bias tool for network meta-analysis. My name is Carole Lunny, and I'm a postdoctoral fellow with the Knowledge Translation Program at Unity Health Toronto, which is an affiliate of the University of Toronto. And I also have affiliations with the Cochrane Hypertension Group and the Therapeutics Initiative at the University of British Columbia. Please tweet me at Carole_Lunny and use the hashtag NMA. So this month, I'm happy to say that we were successful in our CIHR project grant to conduct this research. As a result, this project is fully funded for 3 years. My co-authors and I declare no conflicts or competing interests. So as I work from home, I'd like to acknowledge the land in which this research was conducted, in Squamish, British Columbia, Canada. For thousands of years, it's been the traditional, ancestral, and unceded territory of the Musqueam, Squamish, Tsalo, and Tsleil-Waututh nations. This land is still home to many Indigenous people from across Turtle Island, and I'm grateful to have the opportunity to work on this land. So this project is run by an international steering committee of international experts in network meta-analysis and risk of bias tool development. The steering committee is comprised of Drs. Julian Higgins, Sophia Diaz, Brian Hutton, Ian White, Penny Whiting, Andrea Trico, R.G. Veroniki, and an adjunct, Dr. Giorgio Salanti, who had to drop out of our study due to resource constraints, as well as Dr. Jim Wright, who is one of ours at the Cochrane Hypertension Group. I am the chair and organizer of the committee. Our working group is comprised of Melanie Colnoisier, Tiffany Kim, Mark John, Simin Nagy, Lillian Chen, Mona Arkmalik-Jad, Franklin Hu, and Aya Kapani. So in this presentation, I'll be talking about our background and rationale, the objectives for the development of the tool, methods for developing the tool, the preliminary results of our first objective, which is the methodological review of items related to bias in NMA, and then our next steps, which are our second, third, and fourth objectives. So as an introduction, conventional meta-analysis pool effects from randomized control trials, comparing only 2 treatments. However, network meta-analysis can compare and rank the effectiveness of multiple treatments for the same condition, not just 2. On the left here, we have a pairwise meta-analysis directly comparing 2 treatments. And on the right here, we have a full network of direct and indirect comparisons. When multiple experimental treatments have not been compared in head-to-head trials, a network meta-analysis can infer the indirect effects of an intervention, which are the red dotted lines here in the network plot on the right. From head-to-head trials of the direct effects of 2 interventions from a common comparator, which is usually, the common comparator is usually placebo. Therefore, we can get an indirect estimate from, say, for example, A versus C, even if they were not compared in a real-world trial. So this is just an example here of a network meta-analysis and patient care. So Lee et al. Recently showed that prosglans could have been identified 7 years earlier as the most effective drug class in lowering intraocular pressure from open-angle glaucoma if an NMA had been done at that time. So network meta-analyses provide evidence to clinicians faster because head-to-head trials, head-to-head RCTs had not been conducted yet. That could have provided direct evidence. So there has been a growing number of network meta-analysis over the past 10 years. Here, this graph shows from 2013 to 2018, the relative increase in network meta-analysis. So from 2013, this study had found 91 network meta-analysis. However, in 2017, they had found 316 network meta-analysis. So what is the importance of assessing reviews with network meta-analysis for biases? So one of the things is that we get the problem of conflicting evidence. So for example, here we see across 28 network meta-analyses on the treatment for rheumatoid arthritis, found that all of these network meta-analyses had different results. So among all of these different 28 network meta-analyses, which one do you actually choose? So currently, choosing from conflicting NMAs would be very arduous for any methodologist, and pretty much impossible without a risk of bias tool for network meta-analyses. So the importance of assessing network meta-analysis for biases also relates to if a review is at risk of bias and inappropriate methods are used, the validity of the findings can be also compromised. So we won't know this unless we're able to assess the biases in a network meta-analysis. So here I'm going to pause and launch a poll to assess your knowledge about the risk of about general risk of bias and quality of systematic reviews. I'll reveal the answers after going over the materials pertaining to these questions in within 5 slides. Okay, so the methodological quality of studies, which is basically how well a study is conducted, is often confused with the quality of reporting or the comprehensiveness of reporting. And so reporting quality or reporting comprehensiveness or completeness evaluates how well methods findings are described in a published study. The methodological quality is about identification of systemic flaws that bias or give limitations in the design, conduct, or analysis of research that may distort the findings. So inadequate literature searches, only searching one database, only having one author selecting studies. So these are things that may limit and introduce bias. So here, risk of bias is analogous to internal validity, and it's an assessment of the systematic review limitations which focus on the potential of those methods to bias the findings. Whereas like a quality assessment or critical appraisal kind of refers to items related to quality of reporting as well as mix of bias. Quality assessments usually mix up the two. So what is bias? So there's 2 elements that affect the validity of findings of a systematic review. So one is external validity, which means the applicability of your results to external situations. And the second is internal validity, or basically how correct your estimate is of the effect you're trying to measure. So internal validity depends on bias, and a risk of bias assessment is an assessment of review limitations which focus on the potential of those methods to bias research findings. So bias is a systematic error or deviation from the truth. That means that your results may be incorrect. So here, the results may overestimate or underestimate the effect of a drug interventions. And so different biases, these differences in risk of bias can help explain the variation in the results of studies. So should you believe the results? And to do that, you need to assess each study for risk of bias. So here I want to be clear that bias or systemic error is not the same as imprecision. So bias research, as I said, bias refers to systemic error, so that multiple replications of the same study would reach basically the wrong answers on average, or the right answers on average. So whereas imprecision refers to random error, which is due to sampling variation. So, for example, the results of studies with small sample sizes and few events are less precise. And so this would result in very wide confidence intervals around the intervention effects. Bias is also not the same as quality of study conduct. So quality of conduct is about how well the study is conducted. So for example, even a well-conducted study can be at risk of bias. For example, so when we're thinking about blinding, when blinding is impossible, for example, when surgical trials, surgeons can't possibly be biased to which surgery they're giving because they need to deliver the surgery. So that trial could be a very well conducted trial, but because of the nature of the intervention, bias can be introduced. And then also, bias is not about reporting completeness or reporting quality. So a study can be very well conducted, it can be free of bias, but that doesn't mean that the authors described or wrote up the publication comprehensively so that they've described everything they've done so that it could be replicated. Okay, so in this slide, I am just going to show you the different types of tools that are out there. I know there's a lot. I know this is like a very busy slide, but I just wanted to give you a snapshot of all the tools out there for systematic reviews with pairwise meta-analysis, which is the second column, and then the possible tools for network meta-analysis, systematic reviews with network meta-analysis. So even though we have like so many tools, all of them have different purposes. And currently, there's no tool to assess the risk of bias in systematic reviews with network meta-analysis. So here we see that there's actually in total 40 tools or more that are used to assess the quality of reviews. So, I mean, the most popular ones to assess the quality of published reviews are AmStar 2. However, we also have the Robust tool, which has a specific purpose to assess the risk of bias in systematic reviews with pairwise meta-analysis. So Robust is a domain-based tool which requires careful reading and thoughtful analysis of the studies to adequately rate the risk of bias instead of just using sort of a simple or a simpler checklist method, such as the OQUAC tool. So I just also wanted to give you an example of other tools for network meta-analysis. So to assess the reporting completeness or the reporting quality of network meta-analysis, authors or researchers or knowledge users, anybody who wants to assess it, a network meta-analysis can use the Prisma NMA tool, which was published by Brian Hutton and who's part of our steering committee. Or you can also use the National Institute for Health Research for healthcare and excellence decision making. So this is NICE, Decision Support Unit, that could also be used. But one thing I wanted to note is almost all of the tools for network meta-analysis have been published probably over like about 6 years ago. And however, the methods for doing a network meta-analysis, the statistics of doing them have evolved greatly over this time. So something that might have been published on network meta-analysis in 2011 really isn't what the recommendations would be in 2021 about how to do a network meta-analysis. So if you are looking for information, please use the most current information out there. So which tool would you use for systematic reviews? Here, I want to emphasize that a lot of tools aren't conducted or developed really well. And they need to have done like validity testing. So that's space validity or internal validity. So 5 tools underwent rigorous development, including testing of reliability and validity. However, due to their very different nature of each of these tools, there's not been conclusive evidence about which one you should use. So the ones that have been used for for systematic reviews are AMSTAR 2, AMSTAR 1, ROBUS, which is again for a risk of bias assessment, OQUAC, and Higgins. So with our new network meta-analysis tool, we want to make sure that it is developed reliably and validly. So here, this is just a look at what AMSTAR 2 looks like. So AMSTAR 2 was developed as an update to AMSTAR 1, and Bev Shea was the main author on that. So the tool contains 16 items, and it can be judged, each item can be judged as a yes, a no, or a partial yes. And then the reviews, in general, when you're finished, you can be categorized as high, medium, or low quality. Whereas the risk of bias tool, it kind of contains a similar approach to the Cochrane risk of bias tool. So it has more of a domain-based feeling with signaling questions where you really have to think about answering a question in one way. You can't just look for an answer in the study and then simply say yes or no. You have to do a little bit more thinking. So there's 4 domains in ROBUS, which is study eligibility, identification and selection of studies, data extraction and study appraisal, synthesis and findings, and finally an overall judgment. And so these questions at the bottom here, you have the option of choosing yes, probably no, or no information. So having chosen these signaling questions, you then make a judgment that indicates if you have concerns about the methods relating to each of these domains. So I'm just going to go over the answers to the poll now. Can you bring up the poll for us, Ciprian, so we can see? So here, there is no difference between the quality of conduct of a study and the reporting quality of the study. So sorry, so there is a difference. So yes, majority of people have said false, so that's great. Why should reviewers include published studies in addition to published studies? So this is a mixture of answers to A to C. So more relevant studies will be found if you do search the gray literature. And this reduces the risk of publication bias and as well increases the likelihood of modeling a true effect of the intervention due to a large sample. So I always tell my students that, you know, one of the best things that you could do when you're doing a systematic review is search 1 million databases and then also like really do a really good gray literature search, because you want to find as many relevant studies as possible, right? To answer your question, your research question. If you have all of the studies in existence, for example, then you'll have a true effect. So question number 3, which of the following tools assess risk of bias in systematic reviews with pairwise meta-analysis? So the right answer was the ROBUS tool. If you haven't actually seen it, I would encourage you to go look at it. And does writing a study protocol use a systematic review, limiting biases from being introduced? And this is actually a bit of a trick question because the availability of a study protocol is definitely the way to go because it proves that you've thought about your methods and you're going to be sticking to those methods as much as possible to minimize publication and outcome reporting biases. A study protocol as well helps us to assess say for example, network meta-analysis, because we can compare the protocol to a published network meta-analysis and we could see if the authors reported all of their outcomes and didn't say, you know, decide to drop an outcome because it was statistically insignificant. Okay, now I'll go over our methods. So to develop, test, and disseminate our tool, we have 4 main objectives. The first one I'll be talking about today in more detail. So our first is to conduct a methodological review to develop a list of items related to biases in reviews with network meta-analysis for potential inclusion in our tool. Our second objective will be to conduct multi-round Delphi surveys with network meta-analysis experts to ask them about which tools they think should be included, which items, should be included in our tool. Our third objective is to pilot test our tool for usability, efficiency, comprehension with different user groups. And then finally, we're going to develop and evaluate a KT strategy and a training plan to be able to disseminate our tool. So again, today we're going to be talking about the first objective in more detail. So our protocol for this first objective was published as a preprint in Research Square. It's also in pre-publication and will be published soon as well. So we had to make, as a steering group, we had to make conceptual decisions about the structure of our tool. So first of all, we decided that we're going to go with a domain-based structure, very similar to the Cochrane risk of bias tool to judge randomized control trials. And our ratings will be low risk of bias, high risk of bias, and unclear risk of bias. We'll also be using signaling questions where yes indicates low concern. And then we will only be talking about biases as they pertain to network meta-analysis only. So we're not going to be, the tool isn't going to be a general tool looking at both systematic review methods and also network meta-analysis methods, because we feel that Robus has done a pretty good job at both that, or AmStar 2, whatever tool you'd like to use to assess the systematic review part. But our tool will only be about the quantitative analysis section, so the network meta-analysis section. So eligibility criteria. So I'll go over the methods that we used. First of all, we wanted studies that describe items to assess risk of bias in reviews of network meta-analysis. And specifically, we included articles describing methods for NMAs, such as tools or checklists or standards for network meta-analysis. We also included guidance documents to see if they had recommended any bias items. Also, any commentaries or editorials. And we searched Medline, the Cochrane Library, Gray Literature, and we also did reference checking. And to be able to combine the items together, we did a qualitative analysis where we split items so that each item only covers a single concept. We also grouped similar items together, and then we also only retained items relating to network meta-analysis. So I'll go through our preliminary results. So our search yielded 3,600 citations, approximately 3,400 of which were excluded at the title abstract phase. And in the end, we assessed 181 articles at full text. Of these, we included 63. So 63 of these, 14 were tools, checklists, or standards. 13 were network meta-analysis guidance documents, such as the Cochrane Handbook chapter on network meta-analysis. 26 were articles relating to bias or methods in NMAs. And 6 were papers assessing the methodological quality of a sample of reviews with NMAs. So when we looked at all of these 63 articles, we ended up with 99 items that the steering committee would go through. So the steering committee then decided which of these 99 items to include, exclude, include but maybe edit or amend. They also said if they were unsure about it or whether it should be excluded because it was a general item related to systematic reviews. So all the items were entered into kind of polls that we're doing right now in Zoom, actually. It's been a great tool for us. And then they were voted on. And we, as a group, as a steering committee, then discussed all of the items that might be either an include or a maybe include, and until consensus was reached. So we have created a list of 14 include amend items, which were organized by topic. So for example, effect modifiers. Okay, so I'm also going to see what you guys want to do. So I'm going to kind of do this process with you. So you're going to decide about whether you think an item should be included or excluded in our tool. So it's kind of like a little bit of a Delphi process, but it's very informal. So here the instructions are. So items having to do with a general systematic review conduct, bias or methods are not included. So for example, the process of double screening studies in a systematic review, that pertains to all systematic reviews. So every systematic review should have 2 people independently screening studies so that you don't miss studies. So anything regarding general systematic review methods are an exclude. Items having to do with methods in network meta-analysis are included. Those methods may introduce bias, they may not, but it's up to you to decide whether an item should be included and excluded, which is a method. And then any item also that you think should be included but needs to be edited in some way so it's worded properly, or whether it's worded so that it pertains to bias, that could also be included for now. So thanks everybody for answering. So I'll just go through the answers now. So was an assessment of the risk of bias quality of the included studies undertaken? And in this case, it is an item related to systematic reviews, but it's about general systematic reviews. And so here, this is a no exclude because it doesn't pertain only to network meta-analysis. So are effect modifiers sufficiently similar across the network? And this is a yes include, because for all the trials that you're including, any effect modifiers across all of the comparisons have to be similar. It's kind of very similar to homogeneity, clinical homogeneity in a systematic review. You want all of your trials to be as similar and have all similar PICO inclusion criteria. So were study characteristics reported in terms of PICO elements? And here again, this is not specific to network meta-analysis, so it's a no exclude. So thanks everybody for doing the poll with us. Okay, so I'll move on. Okay, so our final list of items was retained, and we had 14 items, as I mentioned. And each of these items will be described and operationalized in an elaboration and explanation document. So the items will then, using those tools, we'll draft a tool, and then we'll enter these items into a Delphi survey and ask NMA experts about them. So that's what we're up to now. And so our next steps, so this will probably happen in the summer, is to launch Delphi surveys and also a stakeholder survey, which I'll discuss in 2 slides from now. So the purpose of the Delphi surveys is to ask experts which bias items must be included in the tool. So here we are going to invite about 50 experts, and they'll be invited by the steering committee members as experts in network meta-analysis and also tool development. So they'll be asked to vote on the inclusion of these 14 questions. The estimated completion time of the Delphi survey will be 30 to 40 minutes, and each item will be rated as important or critically important. And those items achieving consensus, which is designed as 70% of respondents saying it should be included, is what our target will be. So what is a Delphi process? A Delphi process is not just a simple survey. So what happens is we invite and recruit the experts, then we distribute the survey to them. We get the results back from the first survey, and we see which items were a yes include, and then which ones were a maybe, and which ones were exclude. So based on that, we kind of modify the tool and the survey, and then we launch it again as a second version, distribute it to the same experts, not different people, same people, and we ask them to make a decision again about our kind of modified items, and then they say include, exclude, maybes again. We look at the results of that second survey and we say, okay, so have we actually achieved consensus with all of the items? If we have, we can stop it right there and then write up our results. And if not, we might do a third survey, a third round of the Delphi and we'll stop there actually. Even if we don't achieve consensus, 3 rounds is definitely enough. So we'll stop at that. We're also going to be launching a stakeholder survey. So the Delphi surveys are really for people who understand like all of the methods and statistics and biases that might be introduced in a network meta-analysis. However, there's lots of people who are going to be users of network meta-analysis, you know, like, or who want to learn about network meta-analysis, students, clinicians, decision makers, you know, funders, people at the WHO or Health Canada that might be using the results of these network meta-analysis. So we want to hear what they have to say about the development of a tool as well. And so we'll be asking them about what type of tool they think would be most useful. We'll also ask any NMA users to look at the list of items that we have, those 14 items, to see if we've actually missed anything or if there's anything that they think should be included. We want to actually engage as many people as possible in this process. And then we'll also, so this stakeholder survey, which will be very short, it'll be 15 questions. We don't want to take up too many people's time, and it will take about 15 minutes to complete. And we'll be launching this in May, pretty much at the same time as the Delphi process. So our third objective is to pilot test the tool for usability, efficiency, and comprehension. So I'll be asking 20 systematic review experts and 20 network meta-analysis authors to use our proposed tool and assess other different types of network meta-analysis to see how the tool is used in practice. So how long does it take them to use the tool? Is there any items that they don't understand? So we want them to comment on comprehension of the items and whether any of the items need to be reworded. We'll also ask them about the structure and how, you know, logical the structure is and whether it's easy to understand as they go along. And so then our fourth objective will be to disseminate the tool and train users on how to actually use it. So to do this, we're going to be developing an evidence-based knowledge translation, dissemination strategy, and also a training plan. So our KT goals are to basically to have awareness of the tool, so get people to know that it's out there and properly use it. So that's the training part of it. So in-person meetings or through Zoom actually will be convened to discuss the results and develop key messages with all types of knowledge users. So we'll also deliver training workshops at various conferences such as the Cochrane Colloquium, the Guidelines International Conference, the Evidence-Based Medicine Live Conference, and others. We'll also be asking organizations if they want us to present it. We'll also be housing the tool on our website. And we'll also be developing lots of KT products such as a 10-minute video, as I said, webinars, and example sheets of how to answer each item. So in conclusion, Risk of Bias NMA tool will provide a tool for training knowledge users in how to assess a review with network meta-analysis for important biases. So knowledge users will be able to assess the NMA validity and applicability in evidence-based decision making, and therefore increasing the uptake of relevant evidence and ultimately helping and improving patient outcomes. So the selection of items is almost complete, and we're going to be developing an elaboration and explanation document. And we'll be launching stakeholder and Delphi surveys, which will be sent out this summer. We'll also eventually be training knowledge users in how to assess a review with network meta-analysis for important biases. And finally, it's really important to think that if a network meta-analysis is at risk of bias, the validity of the conclusions can be compromised, just like any network meta-analysis, and can lead to wrong healthcare decisions. So we really need a tool to help us do that. So thank you. I'm ready to take questions.
So thank you, Carole. There are some questions in the chat, and please feel free to enter more questions in the chat. We'll have a few minutes to answer them. So the first one is, well, it's more of a comment, but maybe you want to comment on it, about having a protocol published. It says it makes you accountable, smiley face. But doesn't necessarily mean you will follow through with the rigorous method you've just described. I don't know if you want to comment on that.
Yeah, that's right, actually. So it's true. I mean, having a protocol, it helps you think about the methods that you will be using, like when you're, doing your review. But, things come up, right? So, for example, your data may not be exactly what you thought it might be. And so it's actually okay to change your methods, once you're conducting the review. However, you have to be very transparent about how you've changed them. So you should have a section, in your written, published review that says these are what we changed from the protocol to the published review because of these reasons, and to provide like a rationale and a judgment as well.
Thank you. Another, Jessica is asking, why did you decide not to search Embase?
Yes. Good question, actually. So we did actually pilot our search in Medline and in Embase, and we found that the precision of our search was just very, very low. So basically that means that we retrieved way too many citations that were irrelevant to our question. So we decided that Medline, the Cochrane Library, and so in our gray literature search, we searched we did targeted searches for, at the WHO websites, a lot of the NICE, the NHMRC websites, these websites that are devoted to for decision making and for policy making, to see if they have any guidance or any kind of checklists for doing NMAs. And we thought that would be a really good place to go from there. We also consulted our steering committee, who are experts in NMA, and they have very good expert knowledge about all the studies out there. So we thought that, okay, well, we're not searching Embase, I think that's a good compromise, because I think we probably have found all the relevant articles.
Thank you. And you may have already touched a little bit on the next question, but Kristen and Iris would like to know more information on your gray literature search.
Yeah, that's right. So I did talk about searching, these targeted websites for decision-making bodies that use network meta-analysis. But I could actually maybe provide, well, we're going to be doing a few more polls afterwards. So I'll put a link into our protocol, and you could take a look in there. We have a full appendix with all the gray literature that we're searching in there as well. But it was comprehensive.
So Bada is asking, hi, Carole. I was wondering whether there might be some overlap between your new tool and the grade NMA approach, which is also in development, as I understand. For example, grade NMA also aims to address the similarity of effect modifiers. The intransitivity is the NMA term that they use, I believe, and coherence between direct and indirect comparisons. Could you perhaps explain any possible differences and similarities between your and the grade approach?
So grade is actually a tool to assess the certainty in a body of evidence. Okay, so that's where we come in here. So here I have the different tools that's up here for assessing systematic reviews and network meta-analysis. You could see grade is for assessing the certainty of body of evidence. So ROBUS, and then our new risk of bias tool is only about assessing the risk of bias in a network meta-analysis, whereas grade doesn't only assess risk of bias, it assesses a whole slew of different components. So for example, I have here in this description of the tool, 5 domains are used for grade, and that's risk of bias, which is, what we're looking at. We're only looking at that, but grade also looks at consistency of the whole body of studies. It also looks at directness, and directness is whether all the studies' research questions match with the actual targeted research question. It looks at precision, which, as I said, precision is not a bias, it's about sampling error. And you'd look at basically the confidence interval to see if a meta-analysis is precise or not. And it also assesses publication bias. Risk of bias also includes publication bias, but it's just one component of that. So yes, our actual network meta-analysis risk of bias assessment can feed into grade, so that for that risk of bias approach, but it's only one component of a grade assessment.
Thank you. Another question from Kristen. She says, also, where can we learn about the progress of your project? And would love to see what you've included so far, the N equals 63 articles.
Okay, great. So we are going to be coming out with the results very soon. We'll have it as a preprint, and I expect to, so we're just kind of, as a steering committee, in this, we're meeting actually the first week of April on the Wednesday. And so we're going to be going through the last, kind of firming up the last of the items and writing explanations for each of those items. And I expect to have the results out in May for this study. So, however, what you could do is you can always email me, and I have, there's people who want to be updated on a regular basis about how we're doing. And so I do a regular update every month to a bunch of people. And so I can put you on the mailing list. So I'll put my, when we're doing the poll, I'll put my email in there, and I can add you to our update list.
A question from Benjamin is asking, is the tool intended to be fully applicable to more specialized types of network meta-analysis, such as IPD, individual patient data network meta-analysis, and network meta-analysis of diagnostic test accuracy studies? Or would further amendments be required to the tool?
Yeah, thanks very much. So right now we've wanted to keep it very simple. And so we only wanted it to be applicable to network meta-analysis. We have, we're thinking about these other applications of network meta-analysis and other types of similar types of analyses as well. But once we have our full list, which is related just to network meta-analysis, we're going to see how many of those items pertain to other kind of networks of meta-analyses as well. So I can't actually answer your question right now, but we're hoping it's going to be inclusive as possible. And then if not, what we'll do is we'll kind of do a different, we'll do kind of subspecialties basically, and develop more specialized ones later on.
Thank you. And this could be the final question. Madeline is asking, how would you bring in a sex and gender and equity analysis into this process?
Okay, so, well, since this is a method study, we have the studies that we can, but as part of the development of the tool, we've tried, so I was the convener of the steering committee, and I made sure that basically there was balance of gender within the steering group itself. And we're going to be doing that same thing with all of the surveys. Although sometimes it's hard, because it is a selection process and people will be self-selecting. But if we see that there's a gender imbalance that's happening in people that are answering the surveys, we can actually send it out to more specific groups who are maybe not well represented in the survey. So we can be doing that as well. And then finally, the actual data analysis itself, as I said, is it's about methods, and so hopefully we won't have any problems with regards to sex data.
Well, thank you very much, Carole. A great talk. I'm sure I speak for many people here. We all learned a lot, and it's obvious that many people are watching with much interest to see how this develops and how the tool comes about. So thank you very much, and maybe we'll bring you back once the tool is completed and
great.
people start using it. Thank you, and thank you all very much. And we look forward to see you next time.
Thank you. Bye, everybody.